Hello, guys. This is chapter 19. This is going to be one of the last chapters of the whole class, but also the last chapter of evolution. And this is just how we kind of organize species and how we chart speciation occurring. And this chapter is just really based on phylogenetic trees and the taxonomical model that Carlos Linnaeus produced. So just, having that in mind, let's just get right into it. All right, so taxonomy, this is just a study of how we speciate or identify different species within the whole biodiversity of Earth. So we have millions and millions of different species. We have to identify them in some way. And taxonomy has actually been around for quite some time. Aristotle and Plato tried organizing species thousands of years ago, and now we have a much more complex and accurate way of organizing these species. But basically, this is just how we organize life on Earth. So Carlos Linnaeus, there was some models of how to organize life on Earth before Carlos Linnaeus came around in the 1700s, but he really starts off taxonomy in the modern sense of science and biology. He was a Swedish scientist, and he was very scientifically literate. And if you remember back, we talked about him a little bit when we talked about evolution. He actually didn't accept evolution. He just thought that his way of organizing his way of organizing life was just the best model out there. And he didn't think that species changed. He actually was quoted as saying, God produced, Linnaeus organized. So he really thought he was placed on this earth to really create a system of organizing species. So here's I like to go through this with my students here in class. This is an electric eel. So what type of organism do you think this is? It's actually not even an eel; it's a fish. So these names can be very misleading. Our next example is a koala bear. You think that it's some type of bear, but it's not. It's a marsupial. Another is the flying fox. A lot of people would think, a fox with wings, but the flying fox is actually a bat. So these are just examples of why we have these misleading names. The mountain chicken, you're probably thinking a mountain or a mountain with a chicken on top, but that's not what it is. It's actually a type of frog. And another example of this is like why we have the like the puma or the cougar or the mountain lion. These are all the same species, but they're they're giving multiple common names for it. And yes, both Pitt and Penn State have the same mascot, but they call it different things. They call it Nittany Lion here in Penn State and the Panther out in Pittsburgh. Now, before Linnaeus came around, or I'm sorry, after Linnaeus came around, there was a lot of different ways to organize species. And you can see here we have the ending species patent, or the way of organizing species in 1998. But basically, Linnaeus organized things into like vegetables or plants and animals. And he came up with something called binomial nomenclature. And this is a two-part naming system that classifies these organisms, and he uses Latin. The reason why we use Latin is because it's a dead language. This means that the actual meanings for words aren't going to change. Excuse me. So, like, if we use the word hot, like you would obviously think of like temperature, but hot also has other terms or understandings for that word. So terms might change based on a language that people are still using. In this binomial nomenclature, names are also going to be italicized, which means they are a little bit slanted. In the genus, you can see here the genus species. This is how we use or we I'm sorry we classify organisms by the genus and species, and they're italicized. The genus is capitalized, species is not, and you can see here sometimes we use the genus. We just put a period after like the G, but not always. All right, so going from the most specific to the least specific, you can see we go species, genus, family, order, class, family, kingdom, domain. I flip this around so you can see how that looks with you can see like the panther here. Um, we go from domain, kingdom, phylum, class, order, family, genus, species, and you probably learned it that way. But just to show you the comparison here, so I don't care which way you learn it. You just have to understand domain is the least specific and species is the most specific. Now, we're going to talk a little bit about Carl Woese in class, but basically he founded the last last major domain, and they added this domain on top of kingdom as a broader classification. So basically, we have bacteria, archaea, and eukarya. Bacteria are obviously just bacteria strains. Archaea are single-celled prokaryotic life, but they are actually more closely related to eukarya than bacteria. You would think that they're prokaryotics; they would be closer to bacteria, but they're not. They're actually on the line of eukarya. So these are the three domains: bacteria, archaea, and eukarya. You can see the three different domains here. You have bacteria, archaea, and then eukarya. We have a more recent common ancestor with archaea than bacteria, and that organism lived around there. So the three different domains, we have bacteria. These are prokaryotic, like I said, they're microscopic, single-celled. Their cell wall contains peptidoglycan. This is just a polysaccharide that's found between the cell walls there. They can be beneficial, they can be harmful, they can be neutral to us, but basically bacteria we already know about. Archaea is the second one, and they are a little bit more unknown. Their cell walls do not contain that molecule peptidoglycan, and they usually live in extreme environments, or they can live in extreme environments. They also live in soils, ponds, oceans, and a lot of other places as well. But yeah, this is just the last. This is just the second domain. Ooh, a really cool fact: no archaea hurts humans. Interesting. Yeah. So one of the places they live is like deep sulfur vents that are very hot, or like the geyser vents in Glacier National Park or Yellowstone National Park. They live in these really weird and extreme environments. And then lastly, this is the most diverse group here. This is what we belong to. This is the Eukarya group. This is multicellular. There are some that are unicellular, and those are the protists. They contain membranes and they have a nucleus. I'm sorry, they contain organelles and they have a nucleus. That's what I meant to say. They reproduce sexually. That's the most common type, and they contain the kingdoms, like I said, the protista, plants, fungi, and animals. You can see here bacteria, archaea, and then eukarya. We have these the kingdoms here: the protista, the plants, the fungi, and the animals. On to Cladograms in 19.3. So basically, in 19.1 and 19.2, we went over just how species are classified and taxonomy in general. We went over the three domains. Now we're going to go over how we map out those domains and how we map out those species specifically. So we use things called cladograms, and this is just like an evolutionary tree. And what this does is it allows us to separate groups of organisms based on these derived or shared characteristics. A clade, this is a common ancestor and all of its descendants. So you can see here, if we have like this whole clade, we have this common ancestor with all of the descendants. I'm going to show you some examples of what a clade looks like, because it's important to know what a clade is. And this is where it gets kind of tricky, and you have to understand it's just a common ancestor and all of its descendants. Yeah, so that's going to be it's a tough concept, and I'm still, even in my vert paleo classes, I'm learning more and more about cladistics and the study of cladograms and just the whole evolutionary tree in general. This is a very cool. I would honestly argue this is my favorite part of evolution, is just these cladograms and phylogenetic trees. Well, a common ancestor, the last organism that is related to two or more species. So I'll give you an example. Like, you and your siblings have a common ancestor of your parents. You and your cousins have a common ancestor of your your grandparents. You and your second cousins have a common ancestor of your great grandparents. So as you go further back, you're related to more and more people, just like we're related to more and more species. And this is an example, by the way, of a cladogram. So a clade, it's really important to know that this is a clade because it's taking in one common ancestor and all of its descendants. This is not a clade because it's not all of the descendants of one specific organism. You can see it's leaving out this one, and this one. This is also not a clade because if you look, we're leaving out this one specific organism. Here's a clade because it's one ancestor and all of its organisms. I'll give you an example. Like, this is like talking about dinosaurs and then birds. You have to be a clade, like that's why people say birds are dinosaurs. It's because like this branch of the evolutionary tree, we did come from, or the birds came from an organism that all dinosaurs are related to. So birds are dinosaurs because they belong to the clade of dinosaurs. All right, with these cladograms, we're going to see ancestral versus derived traits. Basically, what these are ancestral traits are these traits are common because of a common ancestor. So you can see here, they have this ancestral state that's going to be found in A, B, C, and D. Because it evolved here, it's going to be seen in all of these organisms. Derived traits are not common in the common ancestor. You can see here, like, this is a derived trait because it evolved later on. So A is not going to have this shared derived trait. Or you can see here, like, it's this unique derived trait is going to be unique to D because it's evolved along the way to D. So A, B, and C are not going to have this trait. So that's the difference here. So ancestral traits basically are they're kind of older, and they evolved in a longer kind of I don't say longer. It's tough to describe. It evolved earlier on, and so all the species have it. Derived traits are less common. They're more unique, if that makes sense. I'm not going to show this video. I'll show it in class. All right, so a phylogenetic tree. This is very similar to a cladogram, but with one major difference. This shows time from the last common ancestor. So this may not show different adaptations evolving, but you're going to see that this shows more time. So you can see here that the cladograms, yes, it shows us evolutionary history and how related we are, but phylogenetics really show time in a scale of some sorts. So you can see here, like, the human and the mouse show a common ancestor until about 66 million years ago. Back here, we have that common ancestor, but with chickens, and it evolved like 300 million years ago. With all of these organisms, we have different time periods when we have the common ancestor, and it's shown in this phylogenetic tree. And you can see this here again, millions of years ago. We have different species, and their speciation rates are different, which means that you're going to have different relatedness between these different species. Going on, skipping all this. All right, node and root. Node, this is a speciation event. Can rotate nodes, same tree, so we can rotate these two, and they're the same phylogenetic tree. A root, that's just the common ancestor there. So you can see this one tree, W, X, Y, Z, we can flip flop here. So along this node, we can flip flop here. We can flip flop even over here, and these are all the same tree. All right, along with these different characteristics, what we see is things called convergent evolution, which means that adaptations are going to be similar within certain species. So you can see this land mammal becoming this porpoise, this land reptile becoming the ichthyosaur, and then the fish becoming the shark. They all evolved very similar adaptations, but differently. Now, all these adaptations make these organisms really good for swimming in the water, but they all evolve differently. So this is a example of what's called convergent evolution. These are almost like analogous structures, if you remember analogous structures. Oh, so and then the first one there. These can be misleading. So originally you might think that dolphins and sharks are very close in nature, but they're actually not. Dolphins are mammals because they evolved to live on land, and when they went back to the water, penguins, even though they have these flippers and a kind of physique that looks like a fish, they're not fish. They're obviously birds. So don't think that just because something has a very similar adaptation means they're very recent in their common ancestry. Behavioral traits such as mating calls can show evolution history as well. So these are it's not just physical; it can be behavioral as well. The molecular clocks. These are really important because they show how close a species is, not just on physical appearance, but more along the lines of like DNA. So basically what they do is they take the genomes of different organisms and they see how closely related certain genes are, and they look at mutation rates. And we can see constant mutation rates just occurring in different organisms throughout the years. And so what we can do is we can better map out phylogenetic trees based on these molecular clocks. And we do this with a bunch of genes, and what you can see here is evolutionary relatedness, or how recent a common ancestor follows along those same lines. So, like between us and chimpanzees, this hemoglobin gene is exactly the same, which we would expect. Like we would expect a human and chimpanzee to have very similar genetics and create a protein that's very similar because we have a recent common ancestor. Same with horses and chimpanzees. I'm sorry, horses and zebras, horses and gorillas, horses or the horse and gorilla. We can see that their evolutionary relatedness is based on how many differences there are in the amino acid sequence. And there is a lot of data that supports this. This is not just like, oh, the anatomical feature looks the same. We have mass amounts of data for this, and it's much more quantitative in nature. So you can see here we have this molecular clock that says, hey, this is about how many mutations occurs, and or per 1,000 or 100,000 or million years, and we can see that based on that we can see how closely related we are to other organisms. And here's the notes on molecular clocks. All right, here's the end of chapter 19.